It's my pleasure now to introduce Erica Andersen-Nissen, who's the director of the Town HVTN Immunology Laboratory at the Hutchinson Cancer Research Institute of South Africa. And she'll be talking to us about innate immune analyses and their importance to understanding correlates of immunity. Go for it, Erica.
Thanks very much for the chance to present to you today. We know that the innate immune system is activated within minutes to hours after vaccination and can modulate the quality and quantity of the long-term protective immune memory responses that lead to protection after a vaccine. And we know that HIV vaccines are detected by the innate immune system by different cell types and through a variety of receptors and pathways, such as those illustrated on this slide. We know that viral vectors such as ALVAC and others are detected through sort of innate antiviral sensing pathways, and that vaccine adjuvants can activate those pathways as well as additional ones, such as the inflammasome pathway that we know Alum is important in activating. And so we really believe that defining innate immune correlates of HIV vaccine immunogenicity and efficacy will allow us to target certain cell types and pathways with future HIV vaccines to improve HIV vaccine potency and durability. So as was mentioned by Georgia and others, there were certain important differences between the RV144 and 702 trials, and one of those was the use of adjuvant in the protein boost, which is very important for activating the innate immune system. So Alum was used in RV144, whereas MF59 was used in 702. And so we want to understand whether the use of those different adjuvants might be important for influencing the vaccine efficacy observed in these trials. This is a busy slide just summarizing some really amazing studies done in non-human primates by Veffa Franchini's group. Others have done really good studies as well, and I'm sure that Rasmi is going to speak about some of the other signatures of protection in her talk in a few minutes. But I just wanted to highlight that Veffa's publication in Nature Medicine in 2016 really showed that there was a difference when you used alum as the adjuvant versus MF59 in a trial of non-human primates looking at the ability of ALVAC-SIV, and the same regimen is used in the RV144 trial to prevent interrectal infection with SIV. And what she found is that alum adjuvant was associated with the delay and onset of SIV MAC251, but not MF59. And I'm highlighting findings related to innate immunity. There are also findings, other studies, other findings from these different studies, but the protection was associated with mucosal NKp44 positive innate lymphoid cells, RAS pathway genes, and other things as well. And subsequent studies in non-human primates have shown that really a CD14 positive monocyte population might be incredibly important for protection, as well as the dose of alum adjuvant is also important to consider. And that's also different between the RV144 and the 705 trial. And so we're really interested in looking at the role of monocytes and K cells and some of the pathways highlighted by these non-human primate studies in the context of HIV vaccination in humans. And so we've been conducting studies within the HVTN over several years now, looking at sort of the early innate immune response to different vaccine vectors and adjuvants. I'm showing you two examples that we've published here. The first was the HVTN 071 trial, where we looked at the early innate immune response to the Merck Ad5 vector, another vaccine trial that did not show efficacy, where we found that there was rapid induction of an antiviral response in genes at 24 hours post-vaccination, and that the day one bulk PBMC gene expression was actually associated with a CD8 T cell response a month later. So there were some really important innate immune responses going on early after vaccination that could predict whether someone would go on to have a strong, at least CD8 T cell response in this case. Then we recently looked at the HVTN 097 trial, which was the RV144 vaccine regimen in South Africa. And in this trial, what we were able to show was that day one serum cytokine induction, as well as bulk PBMC gene expression, was associated with ADCP responses at month 6.5, all the way 2 weeks after the fourth vaccine in this trial, as well as CD4 T cell response magnitudes at month 6.5 and even all the way out to month 12, suggesting that those early events can really affect the downstream adaptive immune and memory responses to the vaccine. And so in thinking about the differences observed in RV144 and 702, we don't have early immune response time points in those trials. And we know based on these other talks and that there are a number of differences between these vaccine trials. So we turned instead to HVTN 107, which was a phase 1 study as part of the P5 vaccine trial and was really designed in response to that original data set from in the non-human primate showing a difference in vaccine effect in whether alum or MF59 was used in the context of the RV144 regimen. And so this human trial was designed to try to compare these adjuvants directly within the context of the 702 regimen. It was a low-risk participants in South Africa, Mozambique, and Zimbabwe. And it was really an opportunity to compare head-to-head. And so here's the schema of the trial shown here, again, with the ALVAC primes and then the protein boost in the context of either a no adjuvant group or alum or MF59. And so we were fortunate to have 18 individuals enroll in each of these groups for this sort of innate immunity subset, where we had the opportunity to collect specimens pre-vaccination and then at days 1, 3, and 7 post-vaccination to really evaluate that early innate immune response and to ask whether those adjuvants could modulate the response to the ALVAC vector, which was clearly going to induce a big innate response that we had shown in the 097 trial. And so what we're hoping to do is to basically test the hypothesis that the use of the MF59 adjuvant altered innate immune responses to the ALVAC regimen in 702 and potentially resulted in a less productive adaptive immune response than seen in RV144 with alum. And so the objective is to assess the effect of these different adjuvants on the phenotype and the transcriptional profile of peripheral blood innate immune cells isolated from participants on that day 0 pre-vaccination after the third vaccination in the trial, and then at days 1, 3, and 7. And so we have cryopreserved PBMCs, and we're proposing to do multicolor flow using a phenotyping panels that were actually currently being used as baseline to measure baseline immune responses in the 705 trial, but we can cross-compare to this trial as well to really examine the relative proportions, phenotypes, and activation states of subsets of monocytes, dendritic cells, NK cells, ILCs, as well as other cell subsets. And then we also have access to RNA stabilized whole blood that was collected in Tempest tubes at these different time points and would like to perform transcriptional profiling by RNA-seq to really determine whether the early signatures of vaccine efficacy described in the NHP studies or signatures that we identified in 097 differ between the adjuvants and may be responsible, or at least partly responsible, for altered adaptive immune system programming. Now, we do have some preliminary data looking at these early time points after the third vaccination, just looking at serum cytokine levels. And what we've found so far is that there really is a kind of different profile when you modulate the innate response to ALVAC with either MF59 or with alum. So here's a graph just showing the fold change in serum concentration of interferon gamma over that first week after vaccination. And you can see in the left with the ALVAC and no adjuvant, we're seeing an induction of interferon gamma, which we also saw in the 097 trial. And then in the center, when we add alum as the adjuvant with that ALVAC-GP120, you see an increase, although it's not statistically significant, but you can see that both the magnitude and the kinetics of the response are really changed when you add the MF59 adjuvant. So we see an initial higher induction of gamma, and it also remains elevated at day 3 post-baseline. And this is a heat map just showing you a summary of all of the different serum cytokines and chemokines that we've measured so far that shows some change post-vaccination in one of these groups. And you can see that the story is really the same for multiple factors, that the MF59 adjuvant really increases that initial response and also prolongs it. And the flavor of the response is slightly different. And so we're encouraged by the fact that we can see a difference in these adjuvants at a systemic immune response level, not looking in mucosa or draining lymph nodes. We're really seeing at a systemic level. And so we hope that this study that we're proposing will really enable us to identify how adjuvants modulate systemic human innate immune responses to a vector prime protein boost regimen, determine whether innate signatures in the alum arm are more highly associated with adaptive response correlative risk identified in RV144, will also enable us to better understand how early innate responses in humans compare with those in non-human primates. You can obviously do much more detailed studies in NHPs, but we know that the pattern of innate recognition in monkeys is different than in humans, and so we can identify where the similarities lie. And I think it will also help us identify innate cell types and pathways that we might target with future HIV vaccines to enhance vaccine efficacy. And I didn't mention in the slides that we also have the opportunity to access a similar time point in the 705 trial. And so it'd be really interesting and important for us to compare the responses seen in the context of the 702 regimen with those seen in the 705 regimen. So I'd just like to thank the many, many people who are involved in the 107 protocol, as well as my laboratory here in Cape Town who did the serum cytokine work and is looking forward to additional studies. Thank you so much.
Thanks, Erica. So we've got a couple questions for you, and I want to kind of do this rapid fire because we have a couple minutes for questions. So there's a question in the Q&A that came up, and the question is asking about comparing inflammatory cytokine responses also in the mucosal samples, and will there be parallel sampling, and would that be one of the proposals to understand what is happening systemically versus what is happening locally at the site of potential transmission?
Yes, that's a great question. And we have mucosal samples from 107, but they focus more on sort of adaptive time points. So we would be able to look at mucosal antibody, particularly antibody responses. We don't have early time point sampling for the mucosal innate response.
What is the coordination typically between the plasma and the mucosal inflammatory, like cytokine response? Is that coordinated, or can that be different? And does that, really explain why you really need to do both independently to really capture the full picture?
I think it's a great question. We haven't looked at it in humans from the sort of the innate response perspective. Georgia spent, a lot of time on the antibody front looking at how those do or do not, in many cases don't correlate so well. But I think it's an open question from the innate side, as well as the kinetics, when you would best catch those things.
Great. Okay. And then there's a complicated question from Stuart that I would hopefully attempt you to answer in maybe more length afterwards. But I want to just like your impression or your rapid response to, he basically is suggesting that, of course, in RV144, the majority of the protection was out in the first 6 months. And since we know, innate shifts can persist for longer than just, days or hours, what is the possibility that protection in RV144 was due to innate immune functions? And could differences between, let's say, testing populations in the RV144 versus 702 maybe account for why there might have been less, protection seen in potentially 702, even if adaptive immune responses were present that we thought were correlates in RV144?
I think it's a great question. And obviously the field of trained immunity is rapidly evolving. And so how those regimens affect particularly monocyte responses that have been implicated in protection in the NHP studies, I think would be great to look at.
Okay, and then one last one, if that's okay. There's one from Susan that I just want to raise. This is from HVTN 049. We learned the critical role of the prime in shaping the immune response following the on M59 boost. Might the differences you are seeing in 107 be more about the interactions of the ALVAC prime and the boost rather than the use of M59 alone?
Yeah, so the nice thing is we have the exact same trials set up for the M59 and Alum arm, so we can directly compare the response at that third vaccination time point. But I think we need to look at that question in the context of comparing to the 097, where we got the response to the initial ALVAC, and maybe that, a lot of responses to the vector might be influencing what we're seeing after subsequent vaccinations.
Thanks so much. Okay, pass it back to Nicole. Thank you, Erica.